Good morning, everyone. This is Lakshmi Chandra, pursuing MSc Microbiology, Central University of Tamil Nadu. As a part of our seminar series, I am going to deal about microbial degradation of environmental pollutants, mainly pesticides. In this seminar, I'll be dealing with these topics. Coming to the microbial degradation is a process in which the microorganisms like bacteria, algae, fungi will involve in the breakdown of complex organic molecules into our simpler substances. This one is a natural phenomena which plays a crucial role in maintaining the environmental sustainability as by waste decomposition, bioremediation, and recycling of the nutrients. In this, through a series of biochemical reactions, microbes transfer the harmful pollutants into a less toxic forms, where the which maintains the ecosystem balance and the healthy conditions of an environment. In this biodegradation process or microbial degradation process will be involved in different kind of processes like bioremediations, soil remediation, biogeochemical cycling. Coming to the pesticides, pesticides are nothing but the chemical compounds which are used to kill or control different kinds of insects, rodents, fungi, weeds, etc. These can be used as a protecting substances for crop fields and agricultural lands. The word pesticides was derived from Latin words, which means the pestis means a black, whereas cadere means killing of the substance or organism. This term was first used in 1925, around 1925, whereas the first pesticide which was used by the ancient Sumerians was elemental sulfur dust, as well as in the 17th century, they have derived a pesticide or a controlling agent, which is highly used as an insecticide from tobacco leaves, which is called as nicotine sulfate. Later in 19th century, they have derived two more natural substances or chemical compounds used as a pesticides from chrysanthemums as well as the from the roots of tropical vegetables. And the first man-made or created pesticide was diphenyl trichloroethane, which was created around last 80 years. Within the 20 years of this dichlorotriphenyl trichloroethane usage, it got banned because of their adverse effects and health hazards in the humans as well as different organisms, etc. Coming to the classification of the pesticides, pesticides have been classified by the WHO based on their toxicity level. They have been classified into five classes: 1A, 1B, 2, 3, and 4. And these are named as extremely hazardous, by highly hazardous, moderate hazardous, slightly hazardous, unlikely hazardous to be in the normal usage. And coming to the environmental fate of the pesticides, pesticides undergo two different processes of processes. One is breakdown process as well as another one is transfer process. In breakdown process, pesticides undergo photodegradation, chemical degradation or biodegradation processes. Whereas in transferring processes, it may undergo that absorption process, which in which the soil absorbs the pesticides when they are sprayed in a different agriculture lands and fields, and they may undergo volatilization process runoff. In this runoff, they pesticides can be transferred from one field to another field, as well as they may uh, uh, mix it with the water flow, and they undergo leaching, and they may be taken by different kinds of organisms and plants, etc. In this environmental fate, the wet transferring process can be done in a two different procedures, like different kinds, like wet deposition and dry depositions, whereas a breakdown process undergoes in a two different forms, like biotic transformation and chemical transformations. In biotic transformations, they may undergo mineralization, in which CO2, H2O, and NH3 are the products which are formed, whereas in metabolization, they may form into a less toxic substances or soluble substances. Whereas in chemical transformation, it undergoes a hydrolysis, oxidation, and reduction reactions. And ultimately, it forms into a depositions of into the soil sediments, water and atmosphere, etc. And they may also be formed into a soil nutrients, as well as they may undergo a biomagnification processes. Coming to the environmental effect of pesticides, pesticides may lead to the air pollution, water pollution, soil contamination in the different fields, as well as the addition to the killing of the insects and weeds. They may lead to the toxic for different kinds of organisms like birds, fishes, beneficial insects, in non-target plants, as well as the humans. In one of the pesticides like aldicarba, which is a highly toxic carbamate insecticide, which shows a non-target effects on humans, and it may lead to the effect on central nervous system of the human beings. And the extensive application of the pesticides may lead to the formation of pesticide resistant insecticides, as well as pesticide resistant weeds formation, as well as pesticide resistant organism evolution. Adverse effects of the pesticides has to be controlled. One of the management system for this controlling adverse effects is microbial degradation process. In this microbial degradation process, bacteria, fungi, algae, or many other microorganisms uptake the pollutants or the pesticide into their cellular system through biosorption process, and the enzymes which are evolved or produced in the cells of the bacteria act on the pesticide molecules or the pollutant molecules and convert them into a less toxic or non-toxic pollutants or any other soluble substances. This soluble substances or non-toxic pollutants or the substances are released into the environment through efflux pumps of the organism. Coming to the microbes involved in the degradation process, first one is a bacteria which metabolize the pesticides and use them as a nutrients and electron donors for the cellular metabolism processes. In this metabolism, there are two kinds of factors which are mainly affecting, like biotic factors and abiotic factors. In abiotic factors, the temperature which around the cells, water availability, nutrient availability, and soil disruption, etc. This biodegradation process converts the highly toxic substances into a less toxic substances of the pesticides. Now, some of the examples for the bacteria are Bacillus, Pseudomonas flavibacterium, Acinetobacter, Arthrobacter, etc. Coming to the fungi, fungi are the most important organisms in the bioremediation of soil. By their mycelial growth, they have an ability to produce extracellular substances in a higher amounts. These extracellular enzymes degrade the substances which are available around them, whether it might be a pesticide molecule or a pollutant molecule or the nutrients which are required for the fungal growth. These enzymes cleave the uh, substances into a simpler forms. By this kind of mechanism, the fungi are able to degrade the pesticides into a simpler forms. For this degradation, the factors like pH of the soil or pH of the environment, minerals availability in the soil or around the or in which the fungi are growing, and moisture content are the factors affecting. Whereas basidiomycetes are the key microorganisms, they are also called as key soil bioremediators, which have an extracellular lignolytic enzymes which converts the toxic compounds into a non-toxic forms. Some other examples are like Fusarium, Aspergillus niger, Penicillium, etc. Coming to the algal species, algae are also one organisms which can be highly involving in the bioremediation process, a key agents of bioremediation process. Algae, which are mostly grown in wet surfaces or soil, moisture areas like water bodies, etc. In this water bodies, the other organisms were much lesser than the algae. For this water remediation processes and all, algae are effectively involved. They can transform the effluent contaminated with the heavy metals, pesticides, organic matters into sources of the nitrogen, carbon sequesters, as well as the production of oxygen. And coming to the actinomycetes, they metabolize the xenobiotic chemicals in soil, water, and adverse environments like acid environments and alkaline environments as well. In the streptomyces, these are the well-researched subgenus of the actinomycetes because of their ability to degrade the pesticides by using different kinds of enzymes like hydrolysis, glycosyl transferases, xylinases, lactases, proteinases, etc. These streptomyces can degrade various pesticide families, including organochlorines, organophosphates, pyrethroids, and urea as well. In this slide, you can see the key enzymes which are involved in the degradation processes are like peroxidases, hydrolases, oxygenases, dehydrogenases, and phosphotryesterases or dehalogenases, etc. And coming to the mechanisms involved in the pesticide degradation. In this, the first step takes place is biosorption or bioaccumulation process, which is nothing but absorption of the chemical pollutants or the substance or the pesticides onto the cell surface of the microorganisms. Later, it got penetrated into the microbial body depending on their molecular structure and of the target xenobiotic. And later, after penetrating, it undergoes an enzymatic degradation process into a simpler compounds, as well as the reactions which are involved in the enzymatic degradation are depicted here. In this, the first reaction undergoes to convert the pesticide or a pollutant into a water-soluble substance through the processes like hydrolysis, oxidation, and reduction reactions. After this, the metabolite which has come as a product is added with the sugars and amino acids, which results in a more water soluble form as a phase 2 metabolite they have mentioned. These phase 2 metabolites, which are more water soluble, are converted into a non-toxic final products. Coming to the examples of each kind of pesticides from each class and their degradation process. For the extremely hazardous pesticides, aldicarb is one of it. This degradation of aldehyde carb can be done by different kinds of bacteria like Arthrobacter, Acinetobacter, Enterobacter, etc. These degradation involves in the two kinds of mechanisms like oxidative pathways and hydrolytic pathways. In this picture, you can see the aldehyde is converted into aldehyde sulfoxide. This aldehyde sulfoxide is converted by the hydrolase enzyme into aldehyde sulfoxide oxime, as well as by aldehyde hydrolase, it was converted into a aldehyde sulfone, which is later converted into aldehyde sulfone oxime. And this aldehyde oximes are later converted into a nitrile molecules, which are as basic substances for the nitrogen source of different microorganisms for their nutrition. Coming to the next class of pesticides, which is nothing but highly hazardous pesticides. Carbofuran is one of the example of it. This carbofuran degradation undergoes in a three main steps. One is hydrolysis of carbamate bond, processing of aromatic fraction, and subsequent degradation of aromatic rings. The carbofuran undergoes hydrolysis, which is converted into the carbofuran 7-phenol ring, which you can see in the picture. This carbofuran 7-phenol ring is converted into 2,8 dihydroxy 8 methyl 6 oxanona and 2,4-dienoic acid in a subsequent hydrolysis reactions, and later and as well as along with the hydrolysis and undergoes in a processing of aromatic fraction. Later, this compound is completely degrading the aromatic ring and forms into a two substances called 3-hydroxy-3-methylbutanoic acid as well as 2-oxopent 4 inert molecules. These carbofuran hydrolase enzymes are coded by the genes called MCD genes, which was cloned from the plasmid DNA of Achromobacter species, which is WM111 strain. The end products which are formed by the carbofuran degradation can be converted into acetyl-CoA and pyruvate molecules, which can be further utilized in the metabolism of the different microbial organisms. These carbofuran degradation bacteria involves like Pseudomonas, Archromobacter, Novus pingobium, etc. Coming to the next one, moderately hazardous pesticides, which is an example of dichlorodiphenyl trichloroethene. In this process, it undergoes different kind of steps. The first one is initial transformation. In this initial transformation, DDT is converted into a two compounds called DDD, which is nothing but 1,1 dichloro 2,2 bis p chlorophenyl ethane, whereas a DDE, which is nothing but 2,2 bis p chlorophenyl 1,1 dichloroethylene. These are the two compounds which are produced in a different kind of conditions. One is like DDD is formed in a anaerobic conditions like oxygen free conditions, whereas DDD is formed when the organisms are grown in a aerobic conditions in the presence of oxygen via dehydrochlorination processes of original DDT. And these both DDD and DDE are the more toxic compounds than the primary compound of DDT. And these DDD is can be further transformed into DDE. Whereas further breakdown of these two triple D and double DE molecules is converted into a DDMU molecules. In this picture, you can see. And this DDMU is nothing but 1-chloro 2,2 bis 4 prime chlorophenyl ethylene, which is again transferred into a different molecules by the hydroxylation reaction by forming by the bacteria like Pseudomonas aeruginosa, which forms a ethanol molecule as DDOH, which can be represented. And this DDOH is further carboxylated into bis 4 chlorophenyl acetate. These DDA is further converted into carbon dioxide molecule as a mineralization reaction. This is the degradation process of dichlorodiphenyl trichloroethane. Coming to the degradation of slightly hazardous pesticides, one of the example is glyphosate. This glyphosate inhibits an enzyme called enyl pyruvyl shikimate 3-phosphate synthase. This inhibition leads to the prevention of synthesizing the aromatic amino acids in the plant cells, which leads to the cell deaths. And the bacteria and fungi, both the microbes, plays a key role in breaking down of these glyphosate molecules. And this glyphosate degradation undergoes in two different kinds of pathways. In the first pathway, dephosphorylation of glyphosate takes place, which is converted the glyphosate into sarcosine. This sarcosine is later converted into a formaldehyde and glycine by the enzyme called sarcosine oxidase through the oxidation reaction. And this formaldehyde and glycine serves as a carbon sources for the bacteria. Whereas coming to the second pathway, in this pathway, oxidation reaction takes place by, for the glyphosate, in which the glyphosate is converted into amino methyl phosphonic acid by the glyphosate oxidase reductase enzymes. And this intermediate amino methyl phosphonic acid is further converted into formyl phosphonate by the dephosphorylation reactions. And this dephosphorylation reaction later undergoes further oxidation into a formaldehyde formation. This formaldehyde is nothing but the carbon source for some of the bacteria. In this, you can see many of the organisms which having a specific genes which are involved in the degradation of pesticide molecules are given. This is from one of the study. And coming to the advantages and disadvantages. Advantages are like this degradation process can be done in on-site, like in situ remediation processes, and this elimination or can be a permanent reaction where from the place where the pesticide can be completely degraded, and it is a cheaper one, and the cost of procedure will be much more lesser than the chemical degradation as well as many other degradation processes. There is a minimize the destruction process of the environment, and it eliminates the long term liability, and it can be coupled with many other chemical techniques as well. And coming to the disadvantages of this degradation are like treatment has to be done in a longer run. It takes a long period of time, and some chemicals cannot be completely degraded or may leads to conversion into a much toxic compounds than the less toxic compounds, which one of the example we have seen in DTT, the first transformation is converted into a DDD and DDE. They are much more toxic than the original form of DDT. So these kind of products may be formed, and the toxicity of contaminants can be the problem of the one of the disadvantages of the degradation. These are the references for this seminar topic. And thank you, everyone. Thank you, Kavya ma'am, for giving me this opportunity. Thanks for everyone.